all, this is Shajata Fatima from the Department of Microbiology. The topic we are gonna see today is the structure of DNA. In this video, we are going to see about what is DNA, the discovery and the structure of DNA, Erwin Chargaff rule, chemical nature of DNA, properties, types, forms and functions of DNA. What is DNA? DNA is nothing but deoxyribonucleic acid. Some type of DNA is found inside the nucleus in the case of prokaryotes, whereas if you take eukaryotes, the DNA is found inside the mitochondria. DNA is a genetic material which encodes for all the living organisms. It is responsible for the transfer of information from one generation to the other generation. So DNA has four major type of macromolecules which are essential for all the forms of life. Every DNA of every single person has unique structures, like how we have unique fingerprints. DNA discovery. James Watson and Francis Crick in the year of 1953 discovered the structure of DNA. Having the lead work of Rosalind Franklin, who visualized the structure of DNA using X-ray crystallography, which lead them for the work of double helix. The structure of DNA is double helix, which looks like a twisted ladder form. The structure of DNA. So you can see the DNA structure here. It is a twisted ladder like form, right, as we said before. So the DNA is said to be a double helical model which is of twisted ladder form. The sides of the DNA is made of a phosphate molecule and the alternative sugar at both the ends. The alternative sugar present in the deoxyribonucleic acid is called as the deoxyribose sugar. The steps you can see in the ladder is made up of a pairs of nitrogen bases which have specific pairing pattern. So the four types of nitrogen bases present in the DNA is adenine, thymine, guanine and cytosine, which pairs specific pattern to form the whole DNA structure. So the adenine pairs with thymine and the guanine pairs with cytosine. This pairing happens because of the amount of the nitrogen bases in the DNA molecule. The amount of adenine and guanine is equal to the amount of thymine and cytosine, which is corresponding to the Erwin Chargaff rule. So what is Erwin Chargaff rule? Erwin Chargaff rule is also called as Chargaff's ratios. It is found by a biochemist called Erwin Chargaff in the year of 1940s. Erwin Chargaff rule states that the DNA from any cell of the organism should be present in 1:1 ratio of pyrimidine and purine bases. The purine content is equal to the pyrimidine content. So here we have four nitrogen bases, right? We separate it to two basically forms, which is called as purines and pyrimidines. The adenine and guanine is called as purines, whereas the thymine and cytosine is called as pyrimidines. So here what is said is like the ratio of pyrimidines is equal to the ratio of purines. So the number of nitrogen bases in the DNA is equal quantities, whereas the adenine is equal to thymine and the cytosine is equal to guanine. So continuing on the structure of DNA, these four nitrogen bases are held together by the hydrogen bonds. Okay, the DNA is comprised of two biopolymer strands which is coiling each other is called as polynucleotides. Polynucleotides are made of simple monomers called nucleotides to form the double helix structure. So each strand has a 5-end group and a 3-end group, whereas the phosphate group is present in the 5-end group, whereas the hydroxyl group is present in the 3-end dash. So these strands of the DNA is called as anti-parallel strands. Why? Because the DNA molecule, if it starts with the 5' end, it ends with the 3' end. Alternatively, if it starts with the 3' end, it ends with the 5' end. So it is called as the anti-parallel strands. So the DNA is composed of deoxynucleotides which are held together by 3-end and 5-end phosphodiester bonds. The alternative strands are held together by the hydrogen bonds, and here adenine and thymine is held together in between. You can see two hydrogen bonds in between adenine and thymine, Whereas when you see in the case of cytosine and guanine, you can find 3 hydrogen bonds present. The chemical structure of DNA. So as we said, the sugar phosphate backbone is present in the DNA, and there are 4 nitrogen bases, which is adenine, thymine, cytosine and guanine. You can find the structure of those 4 nitrogen bases here. And what holds the DNA structure together? It is nothing but the hydrogen bonds which holds the DNA together. You have hydrogen bonds. Adenine and thymine is said to have 2 hydrogen bonds, and the guanine and cytosine is said to have 3 hydrogen bonds in between. So you can see 2 process here, which is the transfer of DNA into RNA is called as transcription process, whereas the RNA, when it's transferred to protein, the process is called as translation process. So you can see the DNA sequencing above. Next comes the hydrogen structure of the DNA helix. It is stabilized by the hydrogen bonding and the hydrophobic interaction between the nitrogen bases. The diameter of the double helix is said to be 2 nanometers, whereas the helix is repeated one after the another in the interval of 3.4 nanometers. You can find 2 grooves in the case of DNA, whereas it is called as the major groove and the minor groove. Okay, there are 2 asymmetric grooves. One is smaller and the other one is bigger than the other one. So the asymmetric grooves are formed as a result of the geometric configuration of bonds between the phosphate and the sugar bases. So what are the asymmetric groove? The grooves are at 120 degrees instead of 180 degrees. The larger groove is called as the major groove and the smaller groove is called as the minor groove. The larger groove and the minor groove are exposed at the edges. So the edges are being used for the analyzing base sequence of the specific DNA molecule. But this process is said to be so crucial. Properties of DNA. So there is a temperature at which the DNA strands completely separate each other is called as, that particular temperature is called as the melting temperature, which is specific for each specific sequence. So the DNA helix can be either left-handed or right-handed. The bDNA with right-handed helix is said to be most stable. So when you heat the DNA sequence, it separates one after other separately, and when you cool it again, it gets hybridized, which means it gets attached. The bDNA with highest melting point have cytosine and guanine content, because the cytosine and guanine content is said to have 3 hydrogen bonds in between, which is stronger. The sequence basis along with the DNA encodes the sequence of amino acids in each protein of the living organism. Next comes the types of DNA. You can find 2 types of DNA in the living organism. One is the nuclear DNA and the other is the mitochondrial DNA. The nuclear DNA is said to be in the eukaryotic organisms like animals, plants and fungi. They store the majority of DNA inside the nucleus. So as the name indicates, it is located inside the nucleus of the eukaryotic cell. The nuclear DNA does 2 copies per cell. The structure of the nuclear DNA is linear and open ends. It includes about 46 chromosomes with 3 billion nucleotides. The DNA is diploid. Nuclear DNA is inherited from 2 parents. The mutation rate of the nuclear DNA is less than 0.3%. The second type of DNA is the mitochondrial DNA. As the name indicates, it is stored inside the mitochondria. It has 100 to 1000 copies per cell. The structure of mitochondrial DNA is circular and closed. It includes 16,569 nucleotides in the human cell. The mitochondrial DNA is haploid in nature. The inherited only from the mother. The mutation rate of the mitochondrial DNA is higher compared to the nuclear DNA. So you can find different forms of DNA here. They are aDNA or A form DNA, bDNA or B form DNA, cDNA or C form DNA, zDNA or Z form DNA. You can find the structure of the different forms of DNA here. which is separated in the form of the number of bases in between. Okay, you can find the structure of DNAs, right? So the most common DNA in the Watson and Crick model is the bDNA or B form DNA. So it is here the listed of features of different kinds of DNA. The helix hardness is said to be right-handed in A and B DNA, whereas it is left-handed in Z DNA. The nucleotide conformation, A DNA is anti-conformation, B DNA is anti-conformation, whereas the Z DNA is said to have zinc conformation in guanine and anti-conformation in cytosine. The base pair placement of aDNA is displaced away from the central axis and it is closer to the major groove. The base pair placement of bDNA is centered over the helical axis, whereas the base pair placement of the zDNA is zigzag pattern in the backbone. So, the base pairing pattern of aDNA is nothing but adenine to thymine, guanine to cytosine. For the bDNA, it is again adenine to thymine and guanine to cytosine. In the case of zDNA, it has alternative purine and pyrimidine contents. The strands orientation are antiparallel for all the types of DNA. The helical shape is said to be a ribbon-like open cylindrical shape in aDNA. It is compact in bDNA, whereas in zDNA, it is long and thin helical shape. The number of base pairs differs on the type of DNA. It is 11 base pairs in the case of aDNA, 10 base pairs in the case of bDNA, 12 base pairs in the case of zDNA. The angle which the DNA is being twisted is 33 degrees in the case of aDNA, it is 36 degrees in the case of bDNA, it is minus 30 degrees in the case of zDNA. Next comes the functions of DNA. DNA has the crucial role in the genetic material in all the living organisms. The replication process, which is the transferring of genetic information from one cell to the other cell, from one generation to the other generation, is the work of DNA during the cell division. Mutations, the changes which occurs in the DNA sequence is referred to as mutations. Major functions of the DNA also includes the cellular metabolism, DNA fingerprinting, gene therapy, transcription, which is the transfer of DNA to RNA, storing genetic information. So the DNA is responsible for the differentiation and evolution. Thank you for your patience on my lecture. For any queries and content related topics, you can mail us at shasciology024@gmail.com. Any emails which is received will be responded within 24 hours of time. A like and subscribe will be encourageable for more videos. Thank you so much.